I hope at FAMU I will be, upgrading myself, whatever I had, I would share and contribute to this society. And in that way, I will advance my knowledge, research. Yes.
So, Dr. Sa, you joined us last fall, is that right? You
No,
did?
this fall.
Oh, this fall. Okay. Which department are you in?
Computer and information science.
I see. I see. Okay. Okay. Wonderful. Wonderful. Well, we can't wait to hear from you. Okay.
Sure. So let me share my slide.
Share screen.
So yes, are you able to see my screen?
I
Yes,
do. I hope
Yes,
I
it's
everything.
very
can.
clear. Dr.
Okay,
Saha, thank you.
great.
That's good.
So um, actually, this is the work I did at USF because I recently moved to FAMU a couple of months ago. So uh, I'm setting up all my lab and everything. Um, I want to present my recent work. This is the recent one where I designed a DeepmapX, which is a deep learning based alignment free pipeline for the DNA mapping. So I'm not sure like how many of you are aware about like uh sorry, how many of you are aware of these DNA mapping things, but briefly I would like to talk about this. It's in simple word, it's like mapping a segment to a reference genome, right? It's like a puzzle piece. Let's say we have a 100 puzzle piece and we want to accurately find out where it belongs in an entire picture. So with that concept, I designed this one based on like deep learning principle, because so far there is no any such tool which actually work with the intelligence. All right, they have a lot of like the most traditional is BWA and BWMEM2 and Bowtie2. Those all are the alignment tools, and those all are based on the Smith-Waterman score and the Needleman-Unz score. So those are like a traditional and a dynamic computational approach. But what I design here is totally based on some intelligence. All right, so I'm going to talk about some background, the architecture of the entire pipeline, what data sets I used, the experimental setup, performance of this pipeline, and the benchmarking results with a clinical sample. So actually this work was in collaboration with USF Genomics program, from where I collected a clinical sample DNA as well. Yes, as I mentioned, like this is something, a borrowed concept from the NLP, and in natural language processing. If we see there is a very well-known problem that is called geographical toponym matching problem. What happens in toponym matching is like let's say you have a city name, but it has multiple names. Throughout the years, the name gets changed, right? So, but ultimately, all these names pointing to the same geographical location, right? So this is called a contextual learning, not the syntax, syntax learning, but it's like a semantics. Semantics is like a meaning. In what sense it can be used. Here is just one example. So in first, I want to fill up here. Apple is my favorite something, fruit. Apple is my favorite, like I work here. It's a company, and Apple is my favorite, maybe like a phone, and I'm going to buy. So in all these cases, Apple is the same word. However, the different context came into the picture, right? In the same way, in context of DNA, the rearrangement, the sequence, uh, the segment, in fact, let's say this segment is not exactly the same one. So this segment and these segments are entirely different. However, the characters, the nucleotides we call, those all are same in both cases, right? The only difference is the arrangement. So these arrangements bring a different context in genomic science. That's why to learn that property in the in genomes, I designed a deep learning pipeline, which is basically a recurrent neural network where it learns again and again and again and again until it achieves its optimal parameter performance. This is my entire pipeline where it has a two half. The left half is a training module and the right half is a mapping module. So first we train the model, okay, the deep learning model. I train using millions of data, and once it trained, that trained model is capable of producing um, the produce. It's a like a it has the ability to uh, transfer learning. So whatever it learn, it transfer, and whenever the new data set come, the uh, learned knowledge is being preserved, and according to that, it work on the new data. All right. So let's focus on the training part, and I will come to the mapping part. Okay. So training model, basically we have a pair of DNA sequence with a label. So it's a supervised machine learning problem where we have a pair of DNA sequence and a label. For example, let's say X1 and X2 with the label like a match or non-match cases. All right. So here the the model training always followed by the embedding, and then after a two layer of the bidirectional gated recurrent unit is the core element of this training part. And once you can ask me like why we need a bi-GRU. So as we know, DNA is a double helical structure, right? So one strand go all the way. If it go like a forward, the other will go in backward. So to preserve and to learn those modules, it has been designed. I mean, it makes a lot of sense to bring that uh, bidirectional GRU model. All right. So with this data, let's say data one is like a pair of DNA sequence which match, okay, and then unmatch. And there are like a series of such cases. It's not like a match or unmatch only, but there are like a moderate cases, right? All those cases we synthesized taking the data from the NCBI and we follow the training process. In the training process, each time the model, the optimal model has been saved as per the performance parameter. Okay. So let's consider right now a performance parameter. I will come back to the performance parameter later. But in a simple term, let's consider a performance parameter, which is when we train with one data set, we got optimum. So we keep training. And once we find that's the best, so that best model would be fine tuned. Fine tuned is a process where the learning parameters in the neural networks, uh, we try to freeze them because that's the optimal value, right? So with the fine tune, what we do, whatever the optimal value of those parameters, we keep it as it is so that that's the best learning can be carried out for the next step. It should not be altered at any cost, right? That's why we do fine tuning. And of course, after fine tuning, let's say the model is totally trained, all right? So how do we test that trained model? We test with the prediction. So this is our blue color is a testing sequence where it is passed through the embedding, and then we directly skip all this process because that is involved for the training. Now we directly come to the trained model because we believe the model is enough trained and the predictions should be great. So this testing value and it predict whether whatever the level it is, y1 and y2, how it is predicted. All right. And and the prediction score determine how robust our model is. So this is just a training workflow. And in that training workflow, of course, all these neural network model work on the different loss function. Since this is a problem of a binary classification, I chose a binary cross entropy loss because that is the most suitable. And yes, the activation function is ReLU, and after the output is like a sigmoid function because we have to choose either match or non-match in a binary classification. And these all are the performance matrices, especially loss, accuracy, precision, recall, and mean average precision. So in short, for this slide, the input data is like a pair of DNA reads with label. Output is a trained model which is able to generate LLVs. Okay, so LLV is it's a learned latent vector. Ultimately, all these sequences is transferred to a vector space, and that vector space I named is a learned latent vector space. And of course, the predicted classification for testing DNA is percentage of accuracy. Now let's talk about the mapping model. Now let's imagine we have a trained model. So what we want to do, we want to test on a new data. Let's consider a real world example. So we have a full genome reference, okay? And we cut into a random pieces. We make a fragment, okay? And then we do embedding and pass through the trained model, which will ultimately produce LLVs for each segments. And it with the indexing, we make a vector database. Now this problem turns to be a vector database search problem. With a new query, let's say we want one query out of them and try to find the best position or best matched entry. So what we do, we just do a search, a vector database search. And from the vector database, it will find the best match. What are the basis of those process? So, there are many vector database search algorithm, and which basically does like it try to find the closest distance between two vectors, right? So in simple word, whatever the closest distance between two vectors, that's the best match in vector space. And that best match, we bring it back in a sequence space where there are like a strings DNA, and then finally we find the best alignment. So it is a problem with the approximate nearest neighbor problem for a given query in a vector database. So for this module, the input is query of DNA read. This is the query, okay? And the output is like the map DNA read on the reference genome. So X was the query and Y prime is the alignment, the, the best mapped segment that has been found from the database. So in that way, it's a mapping problem. Uh, uh, so far, if you have any question, you can just raise it, okay? Are different lengths. As I mentioned, like we can have, sorry. So basically the DNA pair length is measured in the base pair. So this one unit is a one base pair because it's a double strand, right? But when we rupture them and separate apart, it becomes nucleotide. So if you see nucleotide, NT or BP, in its meaning is the same length. So 1, 2, 3, 4. So this is a 4 base pair together. And if we separate them, it is like a 4 NT nucleotide as a size. So I have like a whole different range of the uh, DNA for the training. It starts with a 153, which is a very short, up to like the 16,000 base pair, which is really, really, really long. And those all sequences, um, I have downloaded a human genome sequence, a reference genome from the NCBI, and then I synthesize all these different data sets. So talking about the experimental framework are like, yes, I developed this in the PyTorch and Python library, Python platform, where I worked on a local workstation, which was provided with the GPU and a high-end processor with the memories, like what, 130 gigabyte. And also it has been performed on the USF GAIV cluster, which is, which is comprised of 145 GPU and 7 terabyte of memory. All right, so let me come back to the performance matrices. Uh, in general, most of the machine learning model is evaluated on the basis of accuracy, precision, recall, F1 score, macro F1, weighted F1, these all are the variation of F1 score, and the map. Map is like a mean average precision. All right. So accuracy and the loss. Okay, loss is not given. Loss is just like a 1 unit minus accuracy is a loss. So this pipeline has been validated with this, with all these performance metrics, and map is particularly very much important when we talk about the transfer learning. All right. So once the model is trained and we want to preserve the optimal values of this parameter, we always check the map, because sometimes even you get like a high accuracy, high precision, high recall, high FN score. And if you are unable to see if you are seeing like a map is fluctuating or like, lower in the lower range, that model will not preserve the will not preserve that learned knowledge. So this map is important, especially for this for the DNA mapping case, because we need that knowledge to be transferred when the model see a new DNA segment. All right, so I would like to present some of the results here. This is the training and evaluation for the 153 base pair result, where, you know, I see that this is the loss. This is for macro F1. This is the accuracy, and these are like a precision and recall. So out of them, except loss, everything should be uh, as high as possible. As high as possible in the sense like if it reaches close to 1, it means the model is the best. And for the loss, if it reaches close to 0, that's the best. All right, so this is my training results, where I found loss is like a extremely almost like 0, accuracy is 99%, precision recall is almost like 100%, and map is 100. And for the same thing, what we do prediction. So basically, let me go back to the, so this one is a training model, okay? And for the same training model, what we do, we predict, all right? We haven't done mapping yet in those results. Yeah, so these results are like testify how good our training model is. So this is the prediction result where I see these values as almost same as the training. This is training, this is prediction. Similarly, for the 200 base pair results, we have more loss comparing to the 153. However, it is acceptable in most of the cases. Similarly, for the 400 base pair length, it seems like model is not converging. However, the loss and accuracy is almost like more than 90%. Yes, accuracy is 91. Loss is a lot, 0.5, 0.4, which is not a good model. And prediction, in case of prediction, we have less loss and more accuracy. However, the curve was like zigzag, so it's not a good model. In that way, I did a lot of training and evaluation for different length, 1000 base pair, okay, 2000 base pair, 4000 base pair, 8000 base pair. Yeah, and this is the 16,000 base pair, where I could see the loss is like minimal if we think about the ultra long DNA segment here with the acceptable accuracy and all these values close to 1. This is really a challenging work in the sequence alignment in domain, because nobody could do like a 16,000 base pair within this limited time. For such a ultra long sequence, if they want to do a traditional approach, it will take a few hours to get this result. These are the summary of all those different length. So this curve describes the loss as uh, lower is better, accuracy higher is better. So that's like for the training loss, and this is the prediction loss, which is basically follows for everything, training and prediction. Now this is a clear image where
Quick question.
yes.
When you're looking at the models that you're increasing the base pairs, it looked like the loss and the accuracy was decreasing. But then when you went over the really long one, it seems to have went back up. Do you have any ideas as to why that
So
occurred?
that was actually, I optimized the model. Okay. I have the old results, which is particularly following the same trend. All right. So after that, I optimize some of the core function in the machine learning pipeline, and then I got that result. So the reason of this robustness is a good learning framework of this pipeline.
No, thank you.
Yeah. So this picture, this picture is something like a kind of summary for entire thing. Okay, as we see, like at the smallest length, we have almost like a zero as loss. All right. uh, and when it get increased, we are getting like a more loss, but it is not like too much, because you see the 0 to 0.25, it's within the range. And 16,000 is something acceptable because the model has been optimized as per the requirement. However, all those different performance matrices like accuracy, macro and precision, and map. So map is a bit consistent. If you see this line is consistent, it means the pipeline that I designed is very much robust, and it holds that property for the transfer learning. Now I would like to show some benchmarking results. So benchmarking results are the data set I have downloaded from the NCBI of different species. So here I would like to present 5 species, all right? E. coli and the cerevisiae. So these all species constitute different categories, different family, like a bacteria, yeast. This is also yeast, some fungi, and this is a parasite. All right. And these family have a different challenges in their DNA. So bringing up all these species for benchmarking is really a challenging work, and you could see very much clearly here. So ML is like my proposed pipeline, and BWA is the closest competitor. Okay, they are used for this sequence alignment work. So in each case, I would see my pipeline performing always well, always great, and also consistent, because if you see the blue has some circle, these are the outlier, and the shape of this what is that called, the triangle up and triangle down. So it seems like it stretched all the way, that it means variance is more. In my case, it's always a short and a robust, which justify the learning is basically essential, and the pipeline could efficiently learn the nature of the DNA. All right. So, the pipeline has 2 important notion. First is the quality, okay? How accurately the pipeline could identify the nearest match, the best match, best segment. And the second one is like how fast, okay? Because nowadays, all these sequence alignment pipeline facing the major issue with the time. They, as long as the DNA length, these tool takes more time. So if, let's say, for the 16,000 base pair, sometimes it take a couple of days to map the entire full genome. Therefore, I would like to analyze my pipeline on the basis of the speed. So if you see here, the deep aligner is my proposed pipeline, and BWA MEM2 is the closest competitor. So in terms of like a end to end, so this is like a end to end result for the user time, and for the, you see that the BWA MEM2, it go exponentially, and if you see the length, it's up to the 4,000 only. It even cannot handle the 16,000. All right. So in this case, the orange one, which is my pipeline, it is almost consistent irrespective of the length. You know what the main reason is? Because all these existing tools, they consider a string matching problem. As the string increase, their matching ability, their all these algorithm complexity increases with the quadratic time, because it's a dynamic programming. But in my case, what I did when I transformed this to vector, so the vector dimension is fixed. Whatever the length doesn't matter with the same vector size. Therefore, it is faster enough to handle any length of DNA than the existing tool, which works with the string and the seed extension method. I break, the breakdown of the time of the deep of my pipeline is like, there are different steps. Yeah. First one is like a tokenization embedding. We do LLV generation and the vector database search, right? All these three different breakdown is pretty consistent irrespective of length. This figure show how robust my pipeline is while comparing with the other renowned tool like Bowtie2, Minimap2, BWMEM2. So these all are like different tools used for different length. For example, the BWMEM is the most efficient tool for sequence alignment of the short read. From the same developer, the Minimap2 is efficient for the longer read, and Bowtie2 is regarded as the most efficient for the longest read. So I compared my pipeline with all of them, and I could see like this blue dots is always consistent in terms of accuracy and the efficiency. However, these are like sometimes other pipeline are sometimes higher, sometimes lower, but nobody is consistent as my pipeline. And this is the result of the clinical sample of the Plasmodium vivax, which is also known as the malarial parasite. In that case, if I see the, there is a matching unit, how do we decide one DNA segment is matched or not? So there is a matching score, which is called Smith-Waterman score, okay? So as higher Smith-Waterman score you are taking, let's say for the 100, if you want to consider 100 as the best match, of course 100 is the best match, right? But in the different tools, that value is different. So in the BWA, they consider even like a 30% of match is regarded as a proper match. Okay, so this graph actually showing how the precision can be improved with our tool. If let's say the precision is 145 for a 153 length DNA sequence, if the precision is high, in that case my pipeline have 25% more precise more precise results than the existing tool. So this illustrates the potential of this pipeline to work on any length of any range of length of the DNA and any species without retraining that. In the existing tool, every time you do sequence alignment, it has to go from the beginning. So concluding my work, this is a part of work actually, because I cannot present all of them, but this is a foundational work which can be easily extended to different domain like a genetic identity, molecular diagnostic, where we can find a genetic disorder, forensic DNA analysis for the DNA sample quickly to map, for the from the crime scene, evolutionary studies, like how those DNA are related, at different point of time, functional genomics, genome assembly, and other like the improvement I can bring up with the BLAST, like a database which is a few billion. All right, so this is like on the testing phase, because that's a very large scale work. And to speed up this process, embracing a parallel processing, and also I'm trying to bring some transformer platform like GPT-4 and DNA Bird to see how these models behave in the same scenario. I have some other ongoing projects, and I would be happy to collaborate with all of us here, whoever interested, especially with the quantum machine learning for DNA reads, or like a generative AI for computational cancer analysis, convolutional neural network for brain EEG analysis. This is particularly important for the epilepsy and Alzheimer's disease research. And in terms of the cyber security of different anomaly detection, like a few cert learning, similarly QML, like the quantum machine learning for smart grid system, in the environment, microbiome analysis, where I'm working with the School of Environment for this. And this is like a memory recall ability as per the digital learning device setting, which is I'm working with my graduate student for her thesis. Thank you so much for everyone, your time, and I would like to take a couple of questions.
Yeah, I have one question, Dr. Sa. So you mentioned about alignment. What's the alignment is referring based on some connections between in the same data set, or is what do you mean alignment in that part of the data?
All right, so in DNA, the sequence alignment is uh, is like something, let's say you found a piece of DNA segment, okay?
Mm-hmm.
And with that DNA segment, you want to find out what species it belongs to, right? So
Oh,
what we do, it's like a kind of a database search. So when we do, let's say we found a book, okay? And when we search book, it will give the most related output, like magazine, a research article, or a newspaper, all those like a similar meaning entity. So in
oh,
the same way, in the DNA, it precisely want to know where this DNA segment belongs in the entire reference.
okay.
That's
Okay.
called a sequence alignment problem.
Okay, got it. Yeah, thank you so much.
Sure.
Okay, I have a question.
Yes, sir,
So
please.
when you get the data initially to analyze, do you do like replication of those DNA sequences? How do you input the data in your analysis?
So yeah, that's a good question actually. There are some tool. So basically what we do, we download a reference sequence from the NCBI, right? And then we cut into pieces. That to prepare a synthetic data, there is a tool that is called like a DWG sim. If you search DWG sim, you will get a very standard command line tool that will help you to cut the DNA data and produce more similar data in a synthetic way. So that tool helped me to curate the dataset first, which is a mixture of all the possible, match, non-match, moderate match, different cases. Did I
My
answer your
okay.
question?
I mean, like, let's say you have a sequence of DNA,
Mm.
a known sequence. Then do you amplify that sequence for your data?
No, I don't.
Okay, that part let's say if I give you unknown, let's say unknown sequence, any unknown sequence,
Mhm.
how do you identify the sequence? Just by using the software?
So far, this pipeline has been tested on the known sequences
Okay.
because what we do, we want to make sure first this pipeline works fine, right? So far in the NCBI, all the reference genome has been identified, and it's like a known reference genome. So for at least for this work, we have taken a known, but from that entire sequence, what we do, we just try to take a small piece and then try to find through this pipeline whether this pipeline able to find that exactly the same location or not, right? If it could find out the exact location, it means this pipeline is robust, what the results shows to us. But for unknown species, it has not been tested yet.
Unknown specimen, unknown sequence. You cannot use unknown sequence. Let's say if I give you random sequence.
Yes, so for that, at least I should be aware, like let's say you gave me X sequence, right?
Yeah.
But also I need to know which species it is, because we need to prepare a database. So right now it's a small scale research where I prepare database for only one species. However, it is expandable. Let's say we have multiple species database, right? So in that case, what I can do, we can just feed that sequence and try to find from the entire database, which include maybe all these species, let's say, right? In that case, it should find the proper location of that species database.
Okay. So,
Yes. So that is called like it's a pangenome work where they put all these reference genomes in one database.
okay. Thank you.
Thank you so much.
May I ask another question, please? So this is mainly used, obviously, for medical diagnostics. Is that right?
Not actually. It has a lot of work in the biology domain. The sequence alignment work is a foundational work in most of the biomedicals. They work for the drug discovery, Alzheimer's disease, computational cancer, genomics, and of course the sequence mapping problem. And there are some more. So this is basically a tool that a lot of people can use it for their own applications.
Okay.
And also it can be like extendable to the protein or like RNA, and the single cell analysis or like other multi-omics analysis. So it's just a foundational work that can be extendable to any extent.
Okay. So is there any specific um, marker or something that has been discovered recently for any specific disease, something really big?
Actually, this work has not published yet.
Okay.
So I don't know. I think the recordings should not be accessible to the public because it's not published yet. So I'm
Okay.
requesting someone not to make it public. But it is under preparation. Hopefully by the end of this year, it should publish. because it has been testing with another species. I mean, so many species.
Right.
But this is a benchmark results that I have shared with you.
Oh, wonderful.
Thank you.
Wonderful. Thank you very much.
Thank you.
I guess it's not really a question I have. It's just that let me put it out there. So one of the things that I have noticed is that one of the new trends is the because I'm assuming the way you're putting your data in is you're putting the letter sequence. Sorry, that's how you're entering your data, letter sequence for the pairs that you're putting into your database. One of the breakthroughs, one of the things that we're seeing right now is that like for example, for conversation, instead of having the conversation be converted to text and then fed into the neural network, what's happening is that the actual sound itself is being fed into the neural network. The idea behind it is that you're losing some context when you're removing the actual, the actual talking of the person and just converting it into text, and also the converting to text itself has chances of there being introducing errors into the whole system. And like for example, if you're just feeding it just the letters, you're losing the folding, you're losing the maybe the energies at the different levels, the ways these things might interact with each other. And that those things can help you identify the sequence and where the sequence should be. And so what I'm just wondering is that do you have, for example, pictures or a different format for the information? And like, for example, you had pictures, you could just convert those into a convolutional neural network and then feed those into a neural network instead. Just maybe an idea for future after you get done with this cycle, something that you may want to consider is that this might help improve the accuracy and the overall speed of the system as well.
Mm-hmm. Thank you so much for your advice. All right. So, is there any further question? Because I need to go to pick up my
So
kids.
yeah, so Dr. Sa, I just have a quick comment.
Yes, please, sir. Yes.
So, well, good to know about your work. I'm glad that it wasn't something like cybersecurity. And
I
it
mean,
was
cybersecurity was like my work since my PhD, but this is
right.
my
No, I
recent
understand.
work.
What I'm saying is that as a chemist, right,
Yeah.
I got this topic a little bit better.
Okay.
But still, like a lot of things, the terminologies on your computer science, I'm actually learning these days on my project, but I'll talk about that later next week. But kind of quick question, which is related to Dr. Erdell's comments, possibly is, when you do these calculations, of course, there are always factors to consider in terms of energy, in terms of configuration, what kind of accuracy do I take? And then what you did for your training and testing purpose, computationally, what is your like computational need resources? What kind of did you do it like supercomputers or like what was the computational resources you needed for this particular project?
So, there are a variety of computational resources I used as the scale up the project. I used like a higher end servers which is capable of like handling millions of data and having like a GPU like A100. And so those are like when it reaches to a higher scale. But when we do a preliminary experiments, like try to set up a proof of concept, it does not require a very high-end computer. Even a workstation having a normal GPU around like a RTX, it should be fine to establish that relationship. If the only problem is, if you have like a more training data, that
Right.
requires your higher configuration. But if you have like a less data, then it should not bother too much with with even a workstation.
Okay, that gives some idea. Okay, great. Thank you.
Yes, sure. I would be very happy to help any of us here to bring out some computational resources configuration, because I know a lot of people, uh, a lot of like different schools, they have a kind of appropriate computational resources where they can appropriately run their work. But there are a lot of resources around us. In fact, recently I have one GPU server, so it is capable of handling at least a few millions of data, because it has like a 2 terabyte of memory and a very high-end processor with a modern GPU. So I would be happy to help if any of your work requires so high scale up computational resources.
Well, when Dr. Lo comes back soon, I hope he will talk about the NRP, the National Research Platform, and the computational resources you may use there. So I think this may be of great help for you too, as well as for everybody else. But I'll leave this to him. And yeah, I'm not really sure who. I think Erdal, I think you're in charge here.
Yeah. I was just going to add to that. The National Research Platform is basically several universities. Their super computers are available for an online network, and so basically you can get to several high-powered GPUs, access basically 24/7. I think like last year when we went to a conference, someone did DNA. They did something similar where they had broke down the DNAs of several different species, and also somebody else did the what they call the thing, the artificial sun and earth nuclear fusion. They use those supercomputers to kind of predict whether or not it will damage the container as they're trying to balance the flow of the energy. Just to say that we as FEMU are part of Nautilus, and so we can help you get on there, get you extra power. But other than that, I think Dr. Sa has to go. He has some kiddos to take care of. So,
Yeah, yeah. I'm sorry for
any
that.
last words?
Yeah.
Any last words?
Yeah. Thank you very much, Dr. Sa, for your presentation. This was wonderful, and we'll definitely get together another time. Obviously, 1 hour is not enough to talk about all these things that we want to. So thanks a lot, and I hope to see everybody next week when we'll hear from Dr. Dangi. Okay, thanks a lot, and have a nice day, everybody. Okay,
Thank you so much for having me and provided me this opportunity.
of course.
Thank you. Have a
Yeah.
wonderful day.
Take care, you too.
Uh,
Bye.